Executive Director of the Brisbane Diamantina Health Partnership, and today we are up to our fourth COVID-19 roundtable. And what we're doing today is actually a bit of a reprise of the first one we did back in March. And so we've got Professor David Paterson with us today, and also Professor Paul Young, who are going to speak. If I can get my slides to transition. Okay. So I'd like to start this morning's session by doing an acknowledgement of the traditional owners. So before we begin, I thought it would be good for us to pause and reflect on the fact that today we're meeting, albeit virtually, on custodial land of the oldest living civilization in the world. So I'd like to start by acknowledging the traditional owners of country throughout Australia and recognise their continuing connection to land, waters and culture. This week is the National Reconciliation Week, and the theme this year is that we're all in this together. So we all have a role to play when it comes to reconciliation, and in playing our part, we collectively build relationships and communities that value Aboriginal and Torres Strait Islander people, histories, culture and futures. So today on this call, we're paying our respects to elders, laws, customs and creation spirits, and I extend that respect to Aboriginal Torres Strait Islander people on the call today. Brisbane Diamantina Health Partners acknowledge the important role that Aboriginal and Torres Strait Islander peoples play in our community. And I think with Reconciliation Week this week, it's really important for us to emphasise that. Look, we're talking about COVID-19 today, and we're going to talk about clinical trials and vaccine development. But I guess this week, some pretty big milestones happened around the world, especially in America in terms of the number of deaths. So I'd actually like us all to pause just for a moment of silence to acknowledge the lives lost and the people impacted around the world due to this novel coronavirus. Thank you. So today, with a combination of some pretty good planning and I think a good degree of luck, Australia is actually in a very good position. And we have a really good opportunity now as we think about moving beyond response into recovery. So today's session is a reprise. As I said, we've picked up not all of the speakers from the first event, but for David and Paul to provide us with an update. So BDHP's role, just a quick ad from our sponsors. Our role is to connect the efforts of individuals, organisations, communities in Queensland for better health. And our partners are health and research and education leaders who combine their strengths, state-of-the-art facilities that we have here in Queensland to solve some of the major challenges. So BDHP plays an important role supporting translational research. And I think in relation to what's happening with COVID-19, the role for the organisation is really important, and we're working very hard. I guess for us, in terms of considering the principles for the discussion, sort of a little line that I came up with for myself a few months ago, thinking about what we could do, was that we would do everything we could to help, but not get in the way. There was so much work the health delivery system really needed to do to focus on the response. And as noted, acknowledge the amazing efforts of our frontline workers, but also the administration people and others that are associated with making the health delivery system work. So we thought that it'd be an opportunity for us to collectively understand the response. And today, we're really working to enable the mission for the Health Translation Partnership here, enabling the integration of innovative research with education and training in clinical care and the highest quality of healthcare for our local and extended communities. Today we're all on Zoom, and we're using the University of Queensland's implementation of Zoom. So this isn't open Zoom that you would just sort of get off the street. We are effectively providing a public broadcast today. So whilst we don't expect everyone from the public to be on, there's no confidential information that we're presenting. And so we're using the security that's built into the system and obviously monitoring who's on the call as we go along. But in terms of meeting etiquette, I'd ask if people could, sorry about that, my phone was off, but my computer was on. We're asking if everyone could please just mute your audio, or your phones in my case. And if you like, you could switch off video. We'll try and maintain the focus of the video on the presenters as we go along. So the other thing we want to do is try and be a bit interactive and foster some discussion. Now, obviously that's difficult. We've got 26 people on the call at the moment, but very difficult to open up the mics, so to speak. but we're using a piece of technology that lets us be a bit interactive. So on that basis, what I'd like you to do right now is if you could go onto your phones or onto your laptops and your computers and open up a browser and go to www.menti.com, MENTI.com. And if you go to Menti, what I'll get you to do is add the PIN number, which you can see at the top here, 127054. So I'll just put that into the chat thread, if I can find it, just so that everyone's got a copy of it. So that PIN again, if you go to menti.com and put in 127054. Now, what you can do then is, I would like you, if you can, to ask this, to answer this question. So we're really interested in who's on the call today. And so what we'd like you to do is, in a couple of words, just describe your primary role and contribution as it relates to COVID-19 at the moment. So if you'd like to just add in your comments there, this will form a little word cloud that'll come up on the screen in front of us live. So we've got prevention, information, safety, clinicians, education, public health, vaccines. This is terrific. Keep them coming in. Program management. Good eye. Program manager BDHP. Excellent. Vaccine. So this little word cloud's forming, and what it'll do is we'll be trapped inside this presentation, and obviously we make the presentation and a copy of this this video available for the folks who couldn't join us today. So this is an opportunity, as I said, to record and understand the people who are on the call today. And in a second, I'll move to the next slide where we'll want to understand what you'd like to get out of today. And that'll help for when Paul and David and I present, or when Paul and David present, and we'll do some question and answers. And we're going to use this tool as well, just to put some questions up on the screen. In the previous sessions, we've been very much in sort of presentation mode, and we've tried to answer questions across the Zoom chat thread, but today is designed to be a little bit more interactive, and both David and Paul will present, but we're trying to have some time for questions, and we use this system as a way of doing that. So thanks for those responses, and I'll just move to the next question. So we're really interested to understand what you'd like to get out of today as well. This really helps to BDHP to understand how to structure these sessions and what we might focus on in the future. Many of you will recall that we've done a number of sessions now. So if you'd like to just add in on the screen there what you're interested in, what you'd like to get out of today, and this will start to come up on the screen as well. Research in Southeast Queensland. Yep, excellent. Updates, exclamation mark. Business as COVID. Very, very good. Very interesting. This is very topical at the moment. As everyone would be aware, all of our health and research education groups are working on the COVID response, and really, I guess, what happens in terms of recovery. A learned individual from within our community mentioned or said to me the other day that this was the kind of Uber disruption in some respects for the health system. So I think there's an important opportunity. Building stronger collaborative relationships. Yeah, how wide is the collaboration? Oh, well, that's a good question. I think that's one of those sort of esoteric ones. The state of the game for vaccine and trials. Look, these are the sorts of things we're going to be trying to address today. So thanks very much for that. Look, what we'll do now is we've done a bit of a warm up, and we'll get into the session proper now. So just a quick recap of where we got to from the first round discussions. What we understood in that session was, and David will talk more about this now, the local multi-arm intervention trial with drug candidates and where that was up to. We also talked about some interesting programs we saw around a behavioural intervention program, working with apps and social tools, tools to aid infection control and public health. We talked about vaccine development, and we're going to get a great update today on that. And we talked a bit about biobanking. So those things are progressing. But today what we'll do is we'll really focus in on a couple of these points. The other thing that BDHP did in response to this is we held a number of webinars. And if you have a look at the BrisbaneDiamantina.com website and our COVID-19 resources, just on the left here, I've got a little up screenshot from that site. We're lucky to have as our strategic advisor, Professor Frank Gannon. Now, Frank is still stranded, if you like, over in Ireland at the moment. But Frank's been working diligently in the background and providing sort of a blog for us on a strategic commentary on what's been going on. And there's some terrific papers, and I reviewed a paper that he'd been developing over the last couple of days, reflecting on the Gattaca movie and the sort of point of care technologies that we're going to need in the future and how far away they are. So Frank's commentary has been really useful. These roundtables, as I said, all the recordings are there. We've got some primary resources that point off to the key websites and AHRs, the NH and MRC, these sorts of things, but also Queensland Government, Queensland Health sites, some funding and grants, the things that were active, making sure that we had that up there, and where our partners were in the news, and some other curated resources. So recordings of the website, some little infographics of what we've learned. So please, if you have a look at that and then give us any feedback. So now what I want to do is we'll move into the main part of the discussion here. And what we're going to do first up is hear about the current COVID vaccine landscape from Professor Paul Young. So I will just introduce Paul as the head of the School of Chemistry and Molecular Bioscience at the University of Queensland. And we're really grateful for Paul presenting in our first session. And today is a great opportunity to hear a bit more about not only what Paul is up to in the partnership he's engaged with, but what's going on around the world. So I'll hand over to Paul now. If you're ready, you have the controls. You just need to unmute yourself, Paul, and bring yourself onto video, and I'll make you the focus.
I don't need to bring myself onto video, but I can't share my screen.
Okay.
It's telling me I cannot.
That's okay. If I stop sharing, you'll come on. Okay.
That will be it. Yep.
There you go.
Done.
Thanks, Paul.
Thank you, and thanks for the opportunity to present to everyone.
We can see your screen. Your voice is a little choppy.
Okay, net is not perfect, but we'll see how we go.
It's good now. Thank you.
Right, so good. David's asked me to just present really an update of where we were at last time, and I suggested that given that there's quite a lot happening globally, that maybe I'd give a very brief overview of where the global vaccine initiative is currently standing. But I thought I'd start with the current state of play, and we've hit the 5.5 million cases around the world, as you can see from this data. There's some debate about what the death rate is, and on this particular site, looking at the figures that they've generated, it's upwards of nearly 7% deaths from those that have been diagnosed. Of course, we don't know the total number of infected cases because we don't know about asymptomatic infections, and obviously there's a lot of activity in that space at the moment as new antibody tests have become available. On the right hand side you can see that the impact of COVID in terms of its global reach compared to previous emerging outbreaks such as SARS, MERS and Ebola, and clearly this is off the scale, and this is a once in a lifetime, a generation, 100 years experience, although many of us have been predicting an outbreak like this for some years. I came across a really interesting infographic of global deaths currently and where COVID stands, and it places in context some of the commentary that is around that this is just another or milder case of the flu. Here's influenza over the last little while, and the impact of COVID overtook in May even malaria cases globally. So this is an ongoing pandemic with significant impact globally. So to the vaccines, this isn't a lesson this isn't Vaccine 101, but I thought I'd start off with just a nice graphic that summarizes the sorts of vaccines that people have been working on. You know, the traditional ones like live attenuated viruses and whole inactivated viruses, so measles and polio equivalent to the live attenuated, and whole inactivated.
Paul, I'm really sorry to interrupt. I really apologize. The screen isn't refreshing. I was wondering, could I get you to stop and start your share again? I'm really sorry to do this.
Yep.
We don't seem to be getting an update.
Oh, that's strange.
Oh, that's better. Now it's going. That's good.
So
Thank you. Sorry to interrupt. Apologies.
No, that's okay. I was in full presentation mode. Maybe I'll just leave it like this if that's better. And
yeah, that's good. We can see it now. Thank you.
Yeah, okay. So I was just saying that there are multiple versions of virus vaccine types now, and particularly with recombinant DNA technology over the last 20 or 30 years, a number of other approaches have been developed as well. And these include, small components of viruses or bacteria, recombinant viral vectors that carry the gene of interest of the pathogen you're trying to target, or DNA or RNA on their own. And I think many people will have heard much about these now because they were the first vaccines for COVID entered into human clinical trials. Virus-like particles where there is an attempt to reproduce what a virus looks like, but in a recombinant fashion, delivering them via other vector systems. And last but not least is just using components of the virus themselves that are the main targets of a protective immune response, recombinant subunit proteins. And this is the approach that we're using, and many others are as well. I thought I'd give you an update. There's multiple dashboards around now, not only on infection rates and daily updates of deaths, infections, etc. But there are also good dashboards on the vaccine process. And one of the best is at the London School of Hygiene and Tropical Medicine. So this is the next few slides are just a couple of grabs off that site. And it's got a real-time analysis of where all the different vaccines are up to. And it may or may not surprise you to hear that there's nearly 200, it might have even gone past 200 candidate vaccine programs underway globally. And those are sort of broken down on that left-hand part of the figure into the preclinical stage. Most of these programs are still in preclinical. You can see that there are 55 different protein subunit vaccines that have been picked up either through publications or through media announcements or whatever. And we're in this group here. Those that have entered clinical trials, there are 12 now that have moved into clinical trials. So this will be a number that's increasing substantially over the next month or so. And these are a summary of all of those vaccines that are in clinical trial when they started in their preclinical stages, and now when they've reached into the clinical stage. So they're all in phase 1. There's 1 or 2 that are talking about being in phase 1-2. And I'm sure also you would have heard in the press that there are a number of these that are being conducted now in Australia. So we heard Inovio going into its phase 1 clinical study in Melbourne just the other day. So there are multiple vaccine types on board. We're not in this list yet. Many of these vaccines, putative candidate vaccines, went into human clinical studies before that preclinical animal studies had been done. We decided we wouldn't do that. We're going to be completing our animal trial studies, both toxicology and also the animal protection studies before we enter. But we're still on track. The last time I spoke to you, we were talking about end of June or early July. Probably in that particular talk, we were leaning towards early July, and we're still on that time frame. Those of you in Brisbane might have heard on Nova in the mornings, they're already calling for volunteers for that particular study. So we are on track. Just to remind you of where we came from, we're using a platform technology that was developed almost 10 years ago in our lab, and we've been working on it ever since. We've applied it to a wide range of different virus types: influenza, RSV, MERS, SARS, measles, herpes, and a number of others, all of them having one thing in common, and that is that they all encode a fusion protein that drives or assists in the entry of the virus into the host cell. And we found that our technology can apply widely across a number of different virus families. We've done up to 10 so far. And because we had that background, when CEPI, the Coalition for Epidemic Preparedness Innovations, came on board in 2017, one of their calls for grants was for those with platform technologies to apply to emerging disease scenarios. And so we submitted that application in 2018, were awarded funding and began in 2019. The challenge that CEPI asked in this particular round was to target a 16-week timeframe from the identification of a new virus and identification of the antigen that one wanted to target to product release for clinical trials. That was a revolutionary concept at the time and still is, but it's one we've pretty much met, and we'll be ready to progress into clinical studies in essentially that 16-week timeframe. As part of our grant application, we did in fact suggest to CEPI that they should stress test us in our final year. The idea behind that was to test the 2 years of development work that we would have put into generating this pipeline of activities and actually put us under a stress and to turn around a vaccine literally within 16 weeks. The idea would be that they would send us a sequence of an unknown virus and we would develop that vaccine. That was supposed to happen next year. So I guess COVID-19 turned out to be our stress test, and that's where we currently are sitting. I'm not going to go into too much detail here. This was a graphic I put together to highlight how we would make that 16-week timeline to the clinical phase trials. And this was pathogen identification as the first stage. We predicted we'd be able to make our vaccine candidate within 4 weeks. Then the preclinical studies would ensue, mouse immunogenicity, safety and toxicology, and would also start process development for scaling up manufacture of the vaccine. And hopefully we'd be ready to go into clinical trials once safety and toxicology had been determined in rats as per normal for most drug and vaccine candidates. The program that we actually had targets to be identified that we were towards. And so what we wanted to do was to target influenza and MERS and also RSV. 2 of those were going to go into human clinical trial in the course of the study. And another was just developing a vaccine that would be ready for implementation into a phase 1 clinical trial. As it happens, the influenza clinical trial was already set in place for June this year. And so what we've been able to do is simply swap out our influenza candidate vaccine for the COVID vaccine. As I said, we had this stress test idea for disease X, and I guess COVID-19 is disease X version 1.0. I don't think I need to land too much on this. Basically, it's to say that within a week or so of, in 13 days, I think it was, of us beginning our program. So let me go back a bit. We were looking at whether we could develop or use the COVID outbreak back in early January as a stress test ourselves. So we'd already begun the process of developing a candidate or lead vaccine on January 10th when the sequence first came out. But it was on January the 21st that CEPI contacted us and asked us to retool essentially towards the COVID approach, with the intention of taking us all the way through past phase 1 clinical trials into the rest of the analysis. So as of early January, we were very much 24/7 on the pathway to developing this vaccine. Just to show you what it is, this is the construct we've designed. This is the SARS spike protein, and this is the clamp that is the core of our patented technology that constrains that protein or that structure in place, and actually is what allows our vaccine to be presenting to the immune system exactly the form of the protein that would appear on the virion surface. And you can see here, this is one of our production runs, and it's a nice homogeneous spread of this particular spike protein held in this particular conformation. And that's the big advantage of this particular technology. So where are we at? I was going to walk you through this, but I can just use my cursor to do so. We've obviously done the antigen production. We, in fact, had our lead candidate within 3 weeks of getting the sequence information, but we spent another week trialing a whole host of additional construct designs. And in the end, we generated in excess of 200 individual constructs and determined their expression, and chose the lead out of that 250 odd constructs. We've completed mouse immunogenicity studies, a number of immunogenicity trials in mice, all of which have shown that we've been able to induce very strong and potent neutralizing antibody at levels much higher than we're seeing in recovered patients. So that's a promising sign. We've developed some medium scale manufacture, and also we've now generated stable cell lines that are able to be scaled up. And there are multiple liter fermenting cultures going on at this moment, generating large amounts of protein. It shows the challenges that we have in progressing this is on the accelerated timeline that we have. Normally, you would have months, or if not years, to develop all of the intimate details of each stage, but we've really only got days and weeks. And as I'm sure people would appreciate, working with a protein in the laboratory is very different from working with that protein at large scale. And we have found some differences between the material produced at large scale versus small scale. So we've had those challenges to get across, but as I said, we've managed to do that, and we're still on target. We're in the middle of the toxicology studies, and they will be complete, finished by the mid to end of June. So we'll be getting that data out. We've also begun our animal protection studies. So we've shifted this up into the 16-week period, and we're testing our ability to, the ability of our vaccine to induce protection in multiple animal models. And the two significant ones are hamsters and ferrets. And we're doing this study in the Netherlands. So again, that data should be coming out in mid-June to the end of June. The other component, which people may have heard of because it got a little bit of press, and that because it required some significant additional funding, which was obtained from the federal government, the Queensland government, and a number of foundations, and also generous contributions from the community. And what we wanted to do was to speed up the pipeline process. Normally, this is the pipeline of activity you will see in the development of any drug or vaccine. Preclinical studies leading to clinical trials, regulatory approval, and then you begin large-scale manufacture. And our idea back in early March was that if we could uncouple that large-scale manufacture, bring it forward, and do some at-risk large-scale manufacture, financially at-risk large-scale manufacture, we would be in a position where we'd have material ready much earlier, and hopefully as soon as we get regulatory approval to deploy that into the community. So we've begun that process already, and I've given you a few hints as to where that currently stands. It's essentially process development, and we've got around a few of the early hiccups and challenges that we met. So we're moving into large-scale manufacture relatively quickly now. So that's pretty much where we stand. We've made good progress, and you know, I'm still pleased that we're on track. Just to highlight the team, and particularly note Keith Chappell and Dan Waterson, who have been co-leaders in driving this whole program. Person who's not on this listing is Professor Trent Munro, who came on as project director. He's also the director of the National Biologics Facility at the University of Queensland, and has a wealth of industry experience in the production of biologics, and he's brought incredible skills and expertise to the group. I guess the other final thing, I was going to show you a few headlines. We've been variously called anything from heroes to badasses to I can't remember what the bottom one was. Three wise men. That's right, I remember that one. But I think that the community involvement and collaboration we've seen that most impresses us has been all of the involvement from school children who've written to us. And this is only a small section of one of the walls outside our lab that we use as inspiration for us whenever we get tired. Kids are sending in drawings of the virus, they're wishing us well. And some of the donations have come from these kids as well. There was one boy in Victoria who sent us $6.70 from his share jar. So it's been absolutely a humbling experience to get this sort of community engagement. And I hope we can deliver on the vaccine in the end. But the trials will tell us that. At the moment, everything is looking extremely promising and on track. And with that, I'll leave it there. Thank you.
Thank you very much. Really great. If you unshare your screen, I'll bring up my screen, and then we might. We've got some time for some Q&A. I was yesterday in Charlotte Street in the city, meeting with Keith McNeil, the Chief Clinical Information Officer for Queensland Health, and one of the members of BDHP's board. And I saw all of there's a similar thing, pictures from school children thanking the folks in the department and the hospitals, and it is amazing. So thank you for that. Look, what I want to do is I'll bring up my screen again because I've got a little way here with this tool. So if you can get your Menti tool available again, what we'll do is we'll just click on that and I'll move this forward. Now, look, there's a way for us to just take some questions here. So if you've got some questions, please put them in. Now, I need to say to everyone, it's pretty obvious, and hopefully you'll all get this. I know everyone who's on the call today. I can see all the participants. So I've got the profanity filter on, but just as a warning, please be careful. Other people are going to see this. So if you've got some questions for Paul, please let me know now, and they'll come up on the screen. I can do a certain degree of moderation here, hence my warning. So I've got no questions yet, but hopefully you're typing away. And we've got time for just a few questions.
Must have been abundantly clear.
It was a really good presentation. Here we go. So Paul, you should be able to see these as well. Have you demonstrated any concerns with the antibody-dependent enhancement of infection with candidate vaccines from Brisbane?
Yeah, it's a clear concern for everyone developing this vaccine, because this was seen in some of the animal model studies with SARS. So that's on everyone's agenda. Most of those studies, there's been quite a lot of work that's been undertaken to try and decipher what went on with those enhanced diseases, and the problem was an eosinophilia that infiltrate into lungs, causing a more severe disease on subsequent exposure after a vaccine dose. And that was related very much to a skewed stimulation of the immune response in a Th2 direction, a particular path of the immune response. And so everyone has been working on developing immunogens and adjuvants or immunoenhancers that skew the immune response to either a balanced Th1 or Th2 or a primarily Th1 response. And that's the way we anticipate we'll get across this problem. Having said that, we need to demonstrate that that's the case. So in our animal studies underway in the Netherlands right now, we actually have part of that study addressing exactly this question. So we should be able to get some fairly concrete evidence, at least in an animal model, that our vaccine elicits a response that doesn't enhance disease. So watch this space, I guess, but we're confident because we do know that we have skewed the immune response into away from what has been shown to enhance disease.
Thanks, I've got another question that's just come through on the Zoom chat as well. What's the expected time frame? I know this is going to be a difficult one, but this is an interesting question because it's what's the expected time frame? And also, will the vaccine cover several viruses or COVID-19 only?
So it will cover COVID-19 only. There's probably going to be a little bit of cross-reaction with other coronaviruses, but the information that's out there of people who've looked at cross-protection is that whilst there's a little bit of antibody cross-reactivity, it's not sufficient to protect. So this would be a COVID-19 specific vaccine, but what I'm comfortable in advising is that it will be a vaccine that will likely cover all of the developing mutations and strains that are developing as it moves around the globe. A lot of the mutations that are being seen in these developing new isolates are not in the spike protein, and when they are in the spike protein, they're not in the majority of the population. So there's one or two mutations that have sort of started to be accumulating within the population, different populations, and they won't impact the ability of this vaccine to cover it. So comfortable that we will cover COVID-19 in all its guises, and probably for some years to come.
Thanks, Paul. Maybe one or two more. So this is another one that'll be, you'll have to get your crystal ball out. I'm sure you've been thinking about this given the presentation, but where do you think the large scale manufacture is likely to take place?
Can I just say, watch this space?
Yep, good enough. Let's move on. Could this fast-tracked approach be considered as a way forward for vaccine drug development outside the stimulus of a pandemic? I guess that's what your program was set up for, but what's your response there?
My response is absolutely. We need to learn from this. We've been having conversations with regulatory authorities who are considering, the pathways that have been established and the timelines that are typical for drug and vaccine manufacturing being, quite frankly, unacceptable. And so I think the whole community is looking at what might happen post the pandemic and post how we've actually approached this. So I think we're learning a lot about how we can speed up. And I suspect that hopefully drug and vaccine development might actually, in general, might benefit from the experiences we've had this year.
Great, thanks very much. Look, I've got one more question that's coming on chat, but very similar to the one you'll see on the screen here. The one on the chat's about really about collaborative approaches to developing a vaccine. And the question here is related, reviewing the global scene, do you think vaccination will provide lifelong protection? So maybe if you can weave an answer together around the global collaboration, but also on the global scene where you think this will go for, well, lifelong protection. I mean, I'll let you comment. You're the expert. Go for it.
Yeah, I mean, that second component is very much a crystal ball one. We won't know the answer to that. The likelihood is with protein subunit vaccines, you don't necessarily get lifelong immunity, and booster responses or booster vaccinations are likely to be required. I mean, who knows what that will be? As I said, the data's not in. Clinical information will lead us down the path of understanding that a bit more. But I wouldn't be surprised if an every 5-year type vaccine approach would be required. There are very few vaccines that afford lifelong immunity. Mostly they're the live attenuated virus types. And in terms of global collaboration generally, it's been extraordinary. I've never been in research for more than 40 years, and I've never experienced the sort of collaboration that is happening across the globe. And it's not just academic collaboration, it's reaches across into NGOs and small biotechs and also large pharma. Everyone is reaching out and helping where and whenever they can to assist in all of the endeavours. I guess possibly one of the more disappointing components of the collaboration component is nationally. I mean, we've collaborated very well, and I think the connections we have with the ANU, with the Doherty and CSIRO, have worked really well, and so those sort of collaborations for us have been great. But what would have been good to see right from the very start is perhaps a national approach to this. There's quite a lot of vaccine candidates, not just ours, here in Australia. I mean, ours is getting a lot of attention, but maybe an early forum, as it were, to actually, or an audit of capacity within Australia would have been a useful venture to maybe have multiple arrows in that quiver that we could be firing at this particular target. I think that's something also to take away from this, that I think into the future it would be great to have a more national collaborative approach towards health intervention in general.
Great. Look, thanks very much. The next question that's there, we won't go for that one at the moment because it's pretty obvious. There's never enough funding, but getting that balance right is going to be important. And I think your last point there, Paul, about getting the national approach, I think if we think going forward about response and recovery, these sorts of investments are going to be critical to be able to respond. And this won't be the only thing that we need to deal with.
And maybe, and if I can make one last comment, I mean, this is actually a part of the debate in the CEPI community and has been for a number of years. And I mean, we realized this was going to be an issue, and that is the one of between pandemics, between emergencies. We were getting funding for 3 years for our particular program where we're trying to stitch together a vaccine pipeline. What happens after that 3 years if we're not funded? Do we lose that expertise? Do we lose that infrastructure? So a phrase that CEPI have come up with is talking about a warm base, that there is a need to maintain a warm base that can easily be put back into action when a pandemic emerges. But I think that is an issue that more broadly reaches out into funding of these sorts of initiatives generally. I mean, I suspect when this pandemic is over, people will refocus on other things, quite rightly, but we need to maintain the skill sets and all of the infrastructure in place such that they can easily be turned on next time. We need to immunise ourselves as a manufacturing community to the next pandemic.
That's really good. I like that example. I like that the metaphor. I hear the sort of a military war metaphor a lot, and I'm not comfortable with that. I think that one is better, immunising ourselves and being prepared. Thanks, Paul. Look, I'll move this on. I want to give the rest of the time to David. So, David, hopefully you're ready and you're able to bring yourself off mute. So, Professor David Paterson is the director of the UQ Centre for Clinical Research at the University of Queensland, and David presented on the vaccine, the clinical trials that he was leading. That was a number of weeks ago now, so it'll be really interesting to hear more from you, David.
Thank you, David. Can you let me share my screen?
Absolutely.
Thank you.
Okay, go for it.
All right. So I've got a question for everybody. Could you please on the chat line tell me who this is? And while you're sort of preparing your responses, I wanted to just sort of go back on a little bit of history. That is that on the same day I was approached by a pharma company about a myeloma drug, which they wanted to repurpose for coronavirus. And the day before, their share price had actually gone up by 40% in one day on the Spanish stock market as a result of that news. And that very same day, a person approached the Royal Brisbane and Women's Hospital Foundation saying they had $250,000 that they wanted to put into coronavirus research. And the CEO of that foundation said to me, you know, are you doing anything? And I said, well, we'd love to do a clinical trial. And I've just heard about this myeloma drug. As it was, the myeloma drug, you know, once you actually looked at the data, this was just complete speculation that it even had any in vitro activity against coronavirus. And at the same time, my colleagues at the Doherty were planning an Australia-wide study, but they only had $40,000 in funding. And so I said to them, well, let's, you know, we've got this little 250,000 at Royal Brisbane. Why don't we see if we can get some funding together to do the your study Australia-wide? And in the first week, we raised $2 million, including an anonymous gift of $1 million, which was rumoured to be from Tom Hanks. But once that rumour got out, Clive Palmer revealed himself as the donor of that money. Now, I can't see whether people who have identified this person, David, has anyone come up with some names?
No, unfortunately, I haven't seen anything. So if they do know, they're keeping it a secret.
Okay, so this is a guy called Louis Lasagna, the late Louis Lasagna. And he was very important in the world of epidemiology, in that he basically sort of came out very strongly that observational data, when it comes to interpreting whether a treatment is useful or not, is actually quite useless and misleading. So, for example, in the current situation, you might look at several thousand patients who had COVID-19, look at what drug they got, and then come to some conclusions as to whether that drug was effective. But, you know, just say you were looking at vitamin C and you had nothing else to give except vitamin C. Well, what you might have done is, as a doctor, when a person was very, very crook, you didn't have anything else to give and you thought, bugger it, I'll give them a go with vitamin C. whereas people who weren't looking particularly sick, you just sort of left without any specific treatment. And so what an observational study may show is paradoxically a treatment may actually show a higher mortality than supportive care. And that was one of Lasagna's sort of major observations, that really observational data in terms of treatment is very misleading, and what you need to do is a randomized trial. And that's really the only reliable way of finding out whether a treatment is effective or not. Lasagna is more famous now for a thing called Lasagna's law. And Lasagna's law is, as soon as you have a well-designed randomized trial and are ready to start the trial, the patients with the condition that you wanted to study disappear. And then, of course, when you finish the study, they come back. And that's been exactly the scenario with this particular study, the Australasian COVID-19 trial, or ASCOT. So it was the study that was initially designed at the Doherty, in which Royal Brisbane and Women's Hospital Foundation raised money for. 70 hospitals in Australia, 10 in New Zealand, 2,400 patients with 4 particular arms. And normally, once you have an idea for a randomized trial, to getting the first patient enrolled takes about 12 to 18 months. In this case, we were ready to go after about 6 weeks. Unfortunately, the 4th and 5th weeks, we were thinking, wow, we'd be able to wrap the study up by the end of May. Unfortunately, of course, our patients just disappeared. So what we've had to do is go overseas. And so we've partnered with the George Institute India. The George Institute, as many people know, is based in Sydney, but has a really strong track record of doing trials, particularly in China and also India. And so we're going to do the study overseas. We will retain Australian sites and pick up whoever we can pick up in Australia, but we fully expect that the vast majority of our patients will be enrolled in India. Now, just going back to the original study design, was that there were going to be 4 arms. The first it being standard of care, no drug with purported antiviral activity. Secondly, hydroxychloroquine. Thirdly, a repurposed HIV drug called lopinavir combined with ritonavir. And the 4th arm, hydroxychloroquine plus lopinavir and ritonavir. Now, since that study design was put out, there's been one randomized trial published on a drug called remdesivir, which is an intravenously administered medication made by Gilead. It does seem to have a reduction in length of illness and therefore length of hospital stay, a trend towards reduction in mortality. And so you could certainly argue that in parts of the world which would have access to remdesivir, that it may actually replace the standard of care. But if we're talking about India, or in fact, if we're talking about most places in Australia at the moment, that medication is not available. Now, the other important issue, of course, has been that there have been a number of observational studies which have suggested that use of hydroxychloroquine is associated with a higher mortality. But I do want to just re-emphasize Lasagna's point that in fact these studies were really observational and therefore prone to many biases. And in all of these observational studies, the final line of the study has been that randomized trials are needed. Despite that, we have opted to have a formal review by Melbourne Health Ethics, and a scientific review committee is being established, which hopefully will make a ruling mid to late next week as to whether we should continue the study with hydroxychloroquine. Ivermectin, which is a repurposed antiparasitic drug, is being considered as a substitute, and really we'll have information about the next direction of the study, as I mentioned, hopefully next week. I just want to finish by talking about some epidemiologic work that's being done at UQCCR, really examining if an effective prophylactic regimen was available, or if an effective vaccine was available, who should be first in the queue to get it? Because I'd like to have Paul's sort of comments about this, but clearly going from finding an effective vaccine to being able to produce millions, if not billions of doses is going to be a very, very big ask. And so what we've been looking at is, firstly, well, if a person got coronavirus, who would be most likely to die? So who are the people we do want to protect? And unfortunately, it's dropped off this on the y-axis, but what is the risk of a person actually getting in contact with coronavirus? And this clearly has a lot of work yet to be done on it, this sort of model as to who should be prioritized. And there's obviously going to be ethical and economic arguments that come into play. But people who have a high risk of dying, or, and it might end up being a high risk of other adverse consequence of coronavirus. For example, the elderly or people with high-risk medical conditions, particularly if they were likely to come in contact with coronavirus, might be the first cab off the rank, and then perhaps people at lower risk, either of an adverse consequence or of acquiring the virus in the first place, and then down to, where do we put young, healthy people who might be healthcare workers, for example? Where do they go on the agenda, and who's really the lowest risk? And this is going to be informed by a variety of large epidemiologic case control studies. This is one published last week in the BMJ of more than 20,000 people in the UK. It looked at, when a person was admitted to hospital with coronavirus, what was their risk of death, and they used a multivariable hazards model. But what I really want to just show you is, even when we're accounting for underlying medical conditions, age is such an important risk factor for bad things happening with coronavirus. And you can see the 80 plus group or the 70 plus group, even 60 plus, almost a 5 times higher risk of death. Whereas if you look at chronic medical conditions, they are only an increased risk of a little bit over 1. And so that's certainly going to be something that's going to come into play in terms of how we assess who is actually going to be the people we prioritize in getting vaccines. So I'm going to stop there. I seem to have lost control. Stop share. Dave, could you, I think you've got control back now, and I'd be very happy to take any questions.
Thank you very much, David. That was really, really interesting, very interesting, and a really good question. You're right, there's this challenge of the manufacturing that Paul was describing, but how do you focus that into the high-risk parts of our population? So really terrific, great progress and great presentation. So we've still got some time to go, and I'd be very keen to get questions from people. So the Menti thread's open again, menti.com, and then put in the participant code 127054, and your questions will start to come up on the screen here. I'll also monitor the chat thread. He says, quickly finding the window. So if you've got questions and you can't get them into Menti, please feel free to do that. David, while you were talking, I quickly jumped onto the interwebs and I found the Journal of Clinical Epidemiology, and I found a great quote and a reference to Lasagna's work, and I popped that into the chat thread. So if we've got any questions for David, this is the time. Okay, we've got one come up on the chat thread, David. I'll read it out for everybody. Hydroxy. So has it really led to increased death rate? Will public participant in trial? Question ends. But I think the question is around increased death rate. Obviously, we've all read quite a few of that, people's response. So maybe a comment on that?
Yeah, so in the UK, there is a large randomized trial called the Recovery Trial, and they have asked their Data Safety and Monitoring Board to have an unblinded look at their data as to whether there was a higher risk of death in any particular arms of their study. And they said no, there was not. And they felt it was safe for the study to continue. But obviously, it's such a political issue. I mean, it's one thing being, the public perception of a person like Clive Palmer, but when a person like Donald Trump promotes a drug, it's sort of natural for anyone who might not agree with Trump politically to be against hydroxychloroquine. So it's a really, I guess, I've never seen this degree of politicization of clinical trials and observational studies. It's really been quite fascinating.
Yeah, thanks, David. So we've got a question. I'm sure you can see it on the screen at the moment. It seems that invermectin would be particularly attractive prophylactic option, example household contacts or HCW. Any ideas on studying in this setting? Not everyone will know what those acronyms and some of those words mean, including a little definition there might help as well. Thank you.
Yeah, so ivermectin is an antiparasitic drug that we use for worm infections. We also use it for scabies, and it's been around for a long time and generally has a pretty good safety profile. An HCW is a healthcare worker. And so, in fact, 2 days ago, Wendy Hoy, who is a University of Queensland researcher and a Royal Brisbane and Women's Hospital renal physician, had a publication in the BMJ on exactly this point. And Wendy's experience or her research area is really chronic renal disease in Indigenous communities. And so there is a lot of work going on at the moment about potentially using ivermectin as a prophylactic medication if a person in an Indigenous community was infected, that contacts immediately go on to ivermectin. So there is a lot of work going on.
Thanks, David. We might take this very last question and then we'll work to wrap it up. So David, hopefully you can see this question on the screen as well, but I'll read it for those who may not be able to read. David, given you have to recruit participants overseas, why would an American biotech company conduct phase 1 trials here in Australia? It's a good one.
I guess phase 1 trials are predominantly for safety and to some degree for dose finding. And so because you're not actually assessing efficacy, it would be reasonable to do a phase 1 trial in Australia. And just as a plug, we do have a pretty good phase 1 industry in Australia, including in Brisbane. And I'd encourage overseas companies to do phase 1 because that shouldn't have anything to do with efficacy.
Great. Thanks very much, David. Really appreciate that. So just to conclude, look, thank you, everybody. Thank you for attending and making time to come to the session today. A really big thank you to Professor Paul Young and Professor David Paterson for their presentations today and a reprise from the session. So thank you for helping us get this started back in March as well. I know we're at time. Really appreciate the folks on the meeting today just to give us a bit of feedback. So maybe one more quick Menti poll if you've still got the screen going. Just some feedback if you have on the presentations today, and if there are any areas you'd like us to be considering going forward. The next month or two, it's going to be a bit of a hectic time for BDHP. We've got some stuff going on, business stuff going on in the background, but look, keen to get your feedback. It'll be included in the slides and we'll make this information available on our website. Great presentations. Yeah, absolutely. Content today was terrific. Thank you. And I should say it has been in the other sessions as well. The content and the presentations we've had from the BDHP community and from the partner organisations is terrific, really showcases the strengths of our system and a focus on the need at the moment. Will we get a regular update on another session regards progress? Yeah, look, I think that's something we'll do. We'll follow up with all of the speakers we've had. If you have a look at our website, we've collated a lot of resource information there. We've got a pretty good list of all of the activity that we know is going on. One of the reasons for me to speak to Keith McNeil yesterday was about where I'm seeing, I guess, opportunity for some collaboration amongst those projects. Yeah, for those who don't know, I'm more in the data innovation sort of technology space and working around consent and genomics and the digital space. But I can see a lot of opportunity for joining things up, and we'll keep pushing that out over our website. If you've got some specific feedback, please feel free to come back to myself. I'd like to thank the speakers. I'd like to thank everybody who's joined the call today. And in particular, I'd like to thank the BDHP team for their support to pull these workshops together. We really couldn't do it without the work that they do in the background to engage our speakers and to support us to put these on. So thank you, everybody. Really appreciate it. Have a lovely Friday afternoon now. I know it's 12:03 and I hope you have a lovely, safe and relaxing weekend. We look forward to another presentation in the not too far distant future. So thank you, everybody. Bye-bye.
Thanks very much, David. Bye all.
Thank you.